Welcome, and thank you all for turning out on this beautiful night. I'm Dave Stonestrom, and before I introduce our speaker tonight, I am happy to announce that on April 28th, our next public speaker will be Sam Droege, and Sam will be talking about our nation's 4,000 species of native bees, and so he'll show a lot of bee picture, amazing photography. It's my great pleasure tonight to introduce my colleague, Marjorie Schulz, who is a research hydrologist here in Menlo Park. Jory, as she goes by, got her degrees in geology, first from Knox College and then from University of Missouri. Then she came west to joined the USGS here in Menlo Park, where she worked in Pacific marine geology looking at manganese deposits. And then in 1991, we were lucky enough to get her up onto land to work with the Water Resources Group. And she will be staying close to the ocean, but on land, taking us on a wonderful travel log up and down the coast and into the soil. So without further ado, here's Marjorie. I would like, sorry for the double clutch, I would like to remind people who, and let people know who haven't been here before, we are video broadcasting this, and so at the end of the Marjorie's talk, when we have question and answers, if you could use the microphone, that way our remote audience can hear your question. And so we have two microphones set up toward the rear of the room. And if anyone has trouble getting to the microphone, I'll just raise your hand and I'll carry a microphone to you. So without further ado, here's Jory Schulz.
Thank you, Dave, for the great introduction. And I also want to thank the crew that puts these together every month. I know that it's something that a lot of people come to every month, and thank you for being here. I'm really happy to share some of our work with you, and I'm excited about it, so I hope that you learn something and you enjoy your time here. So we're going to be looking at marine terraces of California. I called it Landscapes from the Waves. It's a little bit of a misnomer. We aren't going to actually just stay on the terraces. We're going to be working on terraced soils. And even though I'm giving the talk and my name was on the flyer, it's taken a lot of people to do this work, and I want to acknowledge all of my colleagues, Corey Lawrence and Dave Stonestrom, who I work with, Art White is a geochemist, Tom Bullen, John Fitzpatrick, everyone has contributed here. And so I'm hoping that you've all driven along the California coast, and maybe while you were driving, you noticed that we have these really flat horizontal landscapes, and Highway 1 just stays on those. And maybe if you look to the east, you notice that it gets hilly and then it gets flat again. Well, these are called marine terraces. This is the landscape we're going to look at today. So here's an overview, and just want to say we're going to look at landscape scale things, but we're also going to go down to the roots and look at the root scale. So we're going to be going from the landscapes down to the roots. Here's our path. I'm going to initially talk about what motivates our work, why we're doing this as part of the USGS. Then I'm going to tell you a little bit about soil development and how these terraces form. You're going to need that foundation to understand what we're doing there. So we're going to run you through that, and it's going to be kind of fun. Then we're going to actually get into some of the data that we've generated in this work on terraced soils, on soil organic matter, that would be carbon in soils, and we're going to end up looking at root processes. And they're all going to tie together by the time we get to the end of the talk. So sit back and relax, enjoy the view. So I'm going to start out here with a big picture thing. This slide shows you pools of carbon, where carbon is on the surface of the Earth. And these numbers are in petagrams of carbon. I had to look this up too. It's 10 to the 15 grams of carbon. These arrows show you the fluxes between the pools. And you see things like the reserves in coal, oil, and gas. That's all the stuff we haven't burned. There's a lot of carbon left there. Deep oceans, These are big pools of carbon, but then the next biggest pool is soils. Soils have like 4 times as much carbon in them as the plants growing on the surface of the earth. And they're almost 3 times as much carbon as the atmosphere. And this is a map of soil organic carbon density, basically where is the carbon on the earth. And right away, I bet you see that it's really dark up here. That means there's more carbon up here on the top. This is the down to a meter depth. When you hear about Arctic problems of the Arctic permafrost melting, this is what they're worried about, this carbon being released back to the atmosphere. But we're not going to be up there. We're going to be down here in the temperate zone. There's not as much carbon, but all of these soils in the temperate zone are what we use to grow our food. And so they're important soils to know, and what's happening in these soils is important to understand. So what we're actually, in the big picture, what's driving our research is a couple of questions. Is soil organic matter at risk for being a source of CO2 under changing climate? Basically, if it gets hotter, if it gets wetter, if it gets drier, is what is stored in that soil as organic matter, the carbon in the soil, will it degrade back to CO2 and become a negative feedback to our problems? And we don't know enough about the soils to answer that yet. But we can also look at it in a more positive way. If we know more about the carbon in the soils, can we increase the amount of carbon delivered to the soil? Can we figure out how to get more carbon back into the soil and keep it there? And if we get it in there, how can we be sure it will stay? And so, the plants are busy fixing carbon out of the atmosphere. Can we use them to help us put that into the ground? Okay, so before we get started onto things, we need to know a little bit about soil development. So here's Soil 101 in about a minute and a half. Soils form at the surface of the earth, and if you look here, this would be, like basic granite. Consider yourself up in Yosemite where it's just a granite. Water is falling on that. It's picking up CO2 from the atmosphere. It's got carbonic acid. It starts to degrade and dissolve that rock. But it also gets help from the plants, and the plants start growing, and they start this process of creating a soil. So this is going from bedrock to soil formation. And soil scientists, when you take a soils class, you learn about the 5 soil forming factors, and that's what these are. And I'm going to go through them real quick. Climate, most of them are pretty, once you think about it, they're like, oh yeah. So if you had this bare rock in the desert and this bare rock in a jungle in Costa Rica, say, where it gets a rainforest kind of thing, you can kind of understand that the rock's going to stay a rock longer in the desert than in the rainforest. We just have more water stuff. So climate is an important factor in the soil forming process. Organisms, so think of that as ecosystems. In the same climate, temperate climate, where you might have a grassland and a forest, the soils that form in those ecosystems are going to look different because of the organisms on top of them. Relief. So that's a geomorphic term. It means topography. Soils forming on a hill slope, they're going to form a little different. Water's going to run through there differently than a soil on a flat surface. So that's a way of saying that the relief, the topography, is going to change the soil properties. Parent material. A soil that forms from a basalt is going to be different than a soil on a granite, which is going to be different than a soil on a limestone. You have different elements as your starting material. And then lastly, time, and that's kind of shown right here. The amount of time you have to make a soil changes how that soil looks. Okay, so there's our foundation. We're going to move on. We're going to come back to this, though. You're going to need to remember this, although I won't test you. Terrace formation. So now, how do we form these lovely landscapes? There's 3 processes you need for marine terrace formation. You need waves. Check, we're on the coast. They create these, as the tides come in and roll out and they move sediment around, you plane off these surfaces. I grabbed this one off the internet just because it was such a beautiful view of what we call bedrock wave-cut surface. we need uplift. We need tectonic activity, movement of the land up. And the third thing we need to create marine terraces is a changing sea level. So when sea level is coming up, where we can undercut sea cliffs, and we erode these wave-cut terraces, these bedrock benches, and you can retreat the sea cliffs. So there's the process of the waves attacking the shore. In California, hopefully if you're here in California, you know about these black lines, which we call faults. And as the faults move, it lifts the land up. So we have uplift on our coast. This is why California's coastline is so much different than South Carolina's coastline. And we know we have changing sea level. Now don't worry, I'm going to run you through this. First thing I want to point out is today is over here. This horizontal axis is today is over here, and it goes back about 450,000 years. This is meters above sea level. So here's the current sea level. This is higher, and then this is lower. And as we go back in time, you can see sea level has changed. And we also happen to know that all of these really low sea levels are due to previous glacial cycles. As the ice sheets build up, they hold the water in them, and the sea levels go down. So this is our last glacial maximum, or the last ice age, the Pleistocene. So in summary, about terraces, we have wave erosion creating a platform. We have lowering sea level, which would abandon that platform, but then we also have this surface land rising above. The uplift is taking that platform, and by the time the sea level comes in again, that platform's so high it doesn't become it's abandoned. So that's how we build these nice flat surfaces, and they're up above the sea level. So this is the sea cliff, the present-day sea cliff in Wilder Ranch State Park, right near Santa Cruz. And I think you can see the features. Here's the bedrock that's called the Santa Cruz Mudstone. And right along the top of there's our paleo wave-cut platform. That's where the waves planed that bedrock down when the sea was high. The other thing that happened is these sediments get deposited on top of that platform. Some of these sediments are deposited while it's underwater, and then there's also beach sediments, and then on top of that you might have alluvial or river sediments. So we have these loose sediments on top of the bedrock. And what I'm really going to be talking to you about today is the soil that forms on those sediments. I just had to use this picture I found on the internet. This is Natural Bridges in 1909, and you can see those features again here. Here's your bedrock platform, here's your sediments, and you can even see the soil on top of that. So now we're going to talk about the soils. But first I want to show you that it's the work we do to get some of these samples that I'm going to show you data from. We do a lot of hand augering to install instruments and to sample soil water. We dig a lot of pits. And we dig and describe and we sample. I did want to point this out to you. When we dug these pits, we were trying to look at some heterogeneity in the soil, and so we sampled every 10 centimeters across this 1 meter wide face and down to 1.5 meters or 2 meters. And I'm going to show you data from that later, so I want you to see how that data was generated from those samples. Okay, so here are two images, again from just north of Santa Cruz. These are some of our sampling sites here. The different colors are the different terraces. When I use numbers of terraces, the lower numbers are always closer to the ocean, and the higher numbers are further away. And I want to point out this first terrace where you drive along on Highway 1, it's not cut by very many streams. The surfaces are bigger, they're less, they're not cut by the streams. And as you go up, they become more and more, God, I just lost that word. And so you have smaller remnants of terraces the further you go up. Now over here I have these ages. The way these have been dated is using the influx of cosmogenic isotopes on the sediment there's a whole talk there. I'm just going to leave it with you. These are minimum ages. We know they're at least this old, but they could be older, slightly older. So you can see that this soil here is at least 65,000 years old. And this soil here, there's the Paleo sea cliff. Do you see that? Here's our present day sea cliff. Here's the Paleo sea cliff. Here's the surface. Here's the next surface. And can you see that Paleo sea cliff is eroded and fallen back? So we have this wonderful stair step of soils. This is actually right here, taken from right here, looking up this flight of soils. The other thing I want to point out is that we've got agriculture on this first terrace, not on these older terraces. As soils form, remember when I talked about how the minerals weather? Down here, there's still nutrients to grow plants. Up here, the soils aren't as productive. They're older and they've lost their nutrients, and they can't farm. Well, they have farmed them. They mostly, if they've ranched them, but that's kind of a hint of where we're going with some of this stuff. So soils on marine terraces are older, further from the ocean. Okay? Here's a picture of those soils. These are soil pits that we dug in Santa Cruz. First thing you probably notice is that they look really different as we go across here. All these dimples, we didn't get a nice picture of that before we started sampling. I'm sorry about that. But we have soil development time here, and what we call these are a soil chronosequence or time sequence, right? So we've got soil development, and these terraces represent these little slices of time where all the soil forming factors are the same. So I'm going to run back through those and tell you what I mean. Climate, these are all forming within like 2 miles of each other. So presumably they've been under the same climatic conditions. Organisms, all of our sampling sites are on coastal prairie. So they all have the same ecosystem currently. I have to tell you that they're all under European annuals, which we know have only been there for 400 years. But the historical record shows that when Europeans arrived, they were coastal prairies. They have the same parent material. Now, we're looking at the sediments on top of those bedrock surfaces, but the sediments have all coming down the streams from the Santa Cruz Mountains and being deposited. They're mostly granitic in composition. So we have the same parent material. They're all really flat marine terraces, so we have the same topographic landscape position. And really then the only big difference between these soils is the amount of time they've been developing or forming, the amount of time they've been sitting there weathering. I also want to say that marine terraces aren't the only place that we get soil chronosequences. There's a lot of work being done on volcanoes where you get lava flows of different ages, all about the same composition. And our friends over at Stanford have been studying chronosequence in Hawaii and getting some really good work out of theirs. And theirs goes over, this is thousands of years, kiloyears. So that goes back to 4 million years. River terraces can be used, glacial moraines, successional beaches or dunes. All of these can be chronosequences, and scientists all over the globe are using these environments to study long-term processes. Here's another old picture I grabbed off the internet. This is the Palo Verde Peninsula in Southern California. If you've ever been there recently, all of this is developed. There's houses up the sides of these hills, but from the 1929, I think, is when this was taken, you can see that there's 6 or 8 marine terraces there. It's a beautiful marine terrace sequence. So what I wanted to say about this, though, is that marine terraces then, because of the soil development and the soil chronosequence aspect, they're this natural laboratory that allows us to study these really long-term processes, things we couldn't replicate in the laboratory. Things like soil development, how carbon builds up, hydrology, water infiltration. There's a lot of other things that we can use these marine terrace natural laboratories for. So now some data. I'm going to run you through it. Don't get anxious. Most of the data I'm going to show you today is going to have depth in the soil on the vertical axis. This one happens to be in meters. We're going from 0 to 8 meters here. So this being the top of the soil, okay? The horizontal axes on all of these particular graphs is the concentration of the mineralogy in the soil. So we took the soil sediments and we did something called X-ray diffraction mineralogy, and it gives us a quantitative amount of the minerals, the different minerals in those soils. Okay, so we've talked about a little bit about primary minerals. Those are the minerals that are in the parent material, all right? Secondary minerals, those are the minerals that form during soil formation. By the end of the talk, I think you'll get an idea of what I mean by that. Excuse me. So silicate minerals and feldspars, does anyone here have a labradorite necklace or something? That's a plagioclase feldspar. That's what's plotted here in red, is the amount of plagioclase feldspar. Iron oxides and clay minerals, those are secondary minerals. Those form as the primary minerals dissolve. And in Terrace 1, you can see we have 40% plagioclase at the bottom and about 30% at the top. So there's already this hint that we're weathering away that plagioclase, that mineral, and we're forming this kaolinite, which is a clay mineral. Okay, so what I mean by weathering it away, if you put salt, which is really a mineral, in water, what happens? It dissolves into the water. It takes a long time to dissolve the silicate minerals, but it does happen with time. And I'm hoping that you're already looking forward with the ages on these terraces. I'm just going to take you down to Terrace 5, where we now have almost 40% kaolinite. So this is a lot of clay in that soil and practically no plagioclase. So in the time of this soil has been forming, we've weathered away all the plagioclase, which is about 40, what had been about 40% plagioclase, and we've formed all this clay. But it took 225,000 odd years. And so as what the soil development also affects is how the water moves through the soil. Now what we have here again is soil depth, and now I've got centimeters rather than meters, and we're going down to 3 meters. This is my first terrace. This is my oldest terrace. And the colors represent the amount of water that during that time of year. So red being dry, purple being wet. And this is time in years. So you can see we dry in the summer, then we have our nice wet winter, and then we dry in the summer. And we did it for a little bit longer time at our older terrace. The thing I want to point out is that the first terrace, we dry out down to 1 meter in the summertime. By the time we get to that 5th terrace, that oldest terrace, when it has all that clay, it really dries only to about 50 centimeters. This is an important thing in how the plants use the water. We know, well, they're European annuals now, but in the past, the native grasses can root down to 3 meters, maybe even 8 meters, depending on where you look and who you ask. But that allowed those plants, they were perennial, mostly of them are perennial, that allowed them access to this deeper water. And when you get clays forming here, that caps off that water and lets it stay deeper for wetter through the summer. Okay, now we're going to move on to the soil organic matter, or carbon in the soil. And here, this is our current work, and I want to just tell you what our research questions have been, and just also a little caveat here. I'm going to show you data. It's a lot of it's preliminary, and my conclusions are going to be preliminary also, but we're going to go through it because it's kind of interesting. So we're going to go back to that. This is now going back to that big picture thing. How is the carbon stored and stabilized in the soil? That's one of the things we want to look at. And what controls the soil carbon change with the depth and through time, through ages. And if you're following with me, I have this in blue to remind you to tell you this is what's going to, hopefully after this, once we look at the full carbon cycle from all of these soils, we'll be able to hopefully model forward in time or backward in time through the chronosequence. We'll be able to use what we know about all the carbon cycling in the soil to actually do some computer modeling. And that would be my colleague, Corey Lawrence. That's not my forte. But anyway, so we're going to look at some of this data. But first, we also have this other great feature in California. Now, this is a precip map of the state. And we've got desert down here, and it's really wet up here. And guess what? We have marine terraces all the way along this coast. That's really cool. This is why here's mean annual precipitation. That's what MAP stands, and MAT is mean annual temperature. The temperatures are all basically the same, which is good because temperature can change how minerals weather. Excuse me. But up here we get a lot more rain. And so what we can do then is use these different marine terrace chronosequences in different precipitation environments and actually do what we call a climo-chronosequence, the climate of the chronosequences. Gives us a really deep look at lots of different parameters. So here's some data. Oh, I should probably go back and say, we have data, I'm going to be showing you data from the Mattole River up here, and that's at the north end of the Lost Coast. If you've ever been up there, it's gorgeous up there. And then data from Santa Cruz. We have just a little bit of data here, and not ready to show any of it. So the Mattole soils have more carbon. Well, here's again soil depth in centimeters down to 2 meters, carbon percent. And you can see that we have way more carbon up north in the soil, even at depth. There's more, in some of it, you could kind of say there's more carbon deep than there is at shallow at Santa Cruz. So that's interesting. There's already a hint that, oh, precipitation makes a big difference. That's cool. We probably knew that. Now here's that data that I showed you we sampled on those grids. This is that carbon, percent carbon, depth with soil, and then the contours is the percent carbon. And so here the orange colors are higher carbon and the blue are lower. And you just see that it goes deeper. It extends way deeper in the Mattole soils than in Santa Cruz. And actually, it wasn't really a surprise to us because when we dug the pits, this is what they looked like up north. These are now the Mattole soils. Those are going to be always MT, and Santa Cruz is going to be S or SCT. These look a lot like our first terrace, but that you can just see that the soils are deep, the carbon runs deeper. We didn't actually need our analyses to tell us that. Sorry. Okay, the other thing we'd measured on these is nitrogen. And so when you do a carbon-nitrogen ratio, again, this is carbon-nitrogen ratio, depth in soil. The blue is the Mattole. You see a big difference here. And we can interpret this as microbial processing of organic matter. Here's what the average bacterial carbon-nitrogen value is. Here's the average plant and fungus carbon-nitrogen value. So the input is like from the plants, right? The plants are growing. That's where we start out at. At Santa Cruz, it looks like the bacteria are chewing up whatever organic material is there, and what we have left are bacterial bodies. So the bacteria are processing. What I mean by that is they're chewing it up, they're processing it, it's higher, it has a lower carbon-nitrogen value. And what happens in that process is that they respire CO2. I'm not going to show you the data, but we have CO2 from these soils, and the CO2 content is about 1.5% at depth in these soils, and in the atmosphere it's 400 ppm or 0.04%. So we have a big difference. So we've got respiration going on from roots, but also, I think, from the microbial biomass down there. And again, here's the carbon-nitrogen values on those grids. Pretty uniform in the northern soils, but we now see some structure, some heterogeneities down here in the Santa Cruz soils. So there's another way to look at the carbon, and that's to use isotopes. Isotope is a way of saying a different weight of the same element. So we have, carbon has three isotopes. Two of them are stable, carbon 12 and carbon 13, and they, in the atmosphere, have a constant ratio. There's a radioactive carbon 14, which I'm sure you probably have heard about. It's formed in the atmosphere as cosmic rays hit the CO2 in the atmosphere. It's taken up by the plants, and it degrades over time, and that's how you can carbon date things, right? So the kind of plant that uses the carbon out of the atmosphere, the type of photosynthesis pathway they use, there's several of them, it sets the ratio of carbon 12 to carbon 13. And we can use that ratio then to follow processes through the soil. It's kind of interesting. Now I've got to let you know here, I'm trained as a geologist. I've been working in soils for a long time, and now I'm going into the plant biology. So it's been pretty exciting and always learning trip. And so if any biologists are out there and I get it wrong, please be nice. Okay, so here's the data on C13, and this is per million. It's a way of ratioing carbon 12 and carbon 13 to a standard. And again, you see differences in Mattole and in Santa Cruz, the brown and the, okay, I guess. Oh. So, but we started up here at about the same value. They're all were coastal prairies, so it was similar plants fixing that carbon. And then again, the microbes are processing it. The microbes change the carbon 12, carbon 13, that they change this value. So again, this data points to microbes using this carbon in Santa Cruz. And they're also going to be using it here, but for some reason, because there's so little carbon, they're able to move, we'll be able to see that shift of their processing the carbon. And we also have C14 data, and this is on the bulk soil. So don't think that this is an age date, okay? It's just what how old that, the age, the fraction modern is what we're going to call it. And that's of the organic material in the soil. And in fraction modern terminology, 1 is the current, it would be current time right now. And anything less than 1 is old. They're pretty similar, and this is a pretty similar trend in a lot of soils. You get, I'm going to say older, but I don't really mean datable older. It's just the carbon down there has been there longer. But at Santa Cruz, again, we have this diversion away from what we see in Mattole. It's older. Something's holding on to it longer there. We have some ideas about this, but they're not ready for prime time. But I think it has something to do with the secondary minerals, those clays I was talking about. So we have less carbon, but the carbon here is older. And so that's a process we really want to focus on. We want to try and get carbon into the soils to stay there. The other thing, I think what was I going to say there? So the soils are really different, and we have that 50% clay. You can't really see it here, but we're going to talk about that later. And so the insights, we have just these preliminary insights. Climate, i.e. soil, water, and wetting, drying. Because, didn't talk to this too much, Mattole, it gets more water, but that water is precipitated over a longer period of the year. Their rainy season starts sooner and ends later than it does down here in Santa Cruz. And also up in Mattole, they have a lot of fog in the summer, so their soils don't dry out as much. So the soil moisture, wetting, drying, seems to be an important component of carbon input and cycling. And then also the carbon, nitrogen, and the C13, they show higher processing of carbon in the Santa Cruz soils. We've been measuring dissolved organic matter and CO2 in these soils, so eventually we're going to have the whole carbon cycle ready, and maybe we'll talk about it when we're there. So the other thing, you've probably noticed that we have some really colorful, beautiful soils here in the older soils in Santa Cruz. That's called soil modeling. And we don't see that up here in Mattole. And we got curious about that because of the carbon content and the age of the soils. Soil models, and I think you can even tell it involves the movement of iron. We've got iron-rich red and yellow spots, and then iron-poor gray zones. These features, if you take a basic soils class, though, you'll probably hear about how these models are indicative of wetland soils, but these are not wetlands, they're upland soils. And we wondered what was going on. And I probably should say a little bit about how iron moves. This is probably really smart crowd, and you already know this, but iron isn't really soluble when there's oxygen around. You need to get low oxygen environments. You need to reduce that iron to get it to soluble and dissolved in the water. Now, these soils are oxygenated all year round. There might be micro pockets of redox, but I don't. We can argue about that if you want to take me on it. So, how do we get these models without reducing conditions? That means low oxygen conditions, without being able to solubilize that iron. Well, one of the first things we thought we'd do was like look at the models and just characterize the percent clay over time using image processing. Fiji, it's free online, it's a great little program. So we started it, our Terrace 1 doesn't have any models, so of course but when you do this image processing and you can quantify it, the gray models are increasing with soil age. So it's a soil development process, something that's happening. And I put this one up because I think you can actually see that gray zone looks very clayey. You can take it up and mush it. You can make ceramics with it. This orange and this red are still pretty sandy. So something has dissolved or weathered this away compared to this. It's not just moving the iron, it's weathering it. That gets me into the plants, because if you look at that, doesn't that look like a root has been there? So I got into some of the root literature, and I learned a lot. If you hadn't taken a botany class in 20 or 30 years, there's a lot of really cool stuff going on in plant physiology and root physiology. What I have learned is that 30 to 60% of the photosynthesized carbon is actually sent down below ground into the roots. And of the carbon that goes below ground, 4 to 70%, depending on the kind of plant, is actually released into something called the rhizosphere. The roots are oozing organic acids. And what's the rhizosphere? The rhizosphere means root sphere. That's what that rhizo means. The region of soil directly influenced by root secretions, exudates, and associated soil microorganisms. It's actually a really important part of the plant processes, the root processes. So there's a lot of carbon going below ground. That's actually pretty cool. Why send carbon into the soil, though? Why would you just make it up here and just put it down there? Well, the root exudates weather the soil. They change the soil chemistry. This was a classic study in 1986 where they used a soil matrix that would change color when the pH changes. Now pH, remember, is acidity. So neutral is 7. Going low is more acid. So you can see that this particular experiment, there's a wall here, and it's one corn plant, and some of the roots are going on either side, and they have the nitrogen available in different forms on either side. And on this side, it had to put out more acidity to grab that nitrogen than over here. And then over here, they have two different kinds of plants, sorghum, which is the grass, and chickpea. And they showed that the chickpea acidifies the soil more than the sorghum. So this is really cool. The plants have a device to acidify the rhizosphere and weather the soil and get the nutrients. The other thing that's really, really interesting when you get into this literature is that they make designer exudates. They need iron. Plants, they don't need a lot of it. It's called a micronutrient, but they have to have it. If you garden and you've ever had to fight iron chlorosis, you know your plants need iron, and usually they give you something to acidify the soil, which can move iron. But in oxygenated environments, it's really hard to get that iron moving. And plant exudates have been designed to grab onto the iron and solubilize it by having it complexed with an organic compound so that the plant can then utilize it. So here's a way to move iron in the soil without it being a low oxygen environment, and the plants have done it. So the roots and the associated microbiota, and there's an amazing amount of really interesting work being done on the symbiotic nature of roots in the microbiota in the root zone. They create these really steep zones of biogeochemical environments where we're going from a pH of like almost 7 to a pH of 4 in a half a millimeter, or I think maybe as much as 5 millimeters. I don't remember now. But anyway, it's an amazing steep biogeochemical gradient that these roots can cause. And so I'm going to show you this little cartoon so you can kind of understand this a little better. Here's our little root. It's exuding organic acids, trying to dissolve things, but that stimulates microbial growth, and they want that. It's something that helps them. The microbes grow, but the microbes also exude things to release nutrients. So they have this shared symbiosis. You know, I'm going to give you some carbon, you're going to help me get nutrients. Okay? And then behind the front of the root is sucking up the water. And this is where they get the nutrients going from. And it creates a leach zone, a zone where those nutrients have been. Now, if you're like me, I used to think of roots as just like the straw, and it would just suck up the water, and the plants were happy. And it's not really that way. They move, they're dynamic. And these fine roots that do most of this kind of work aren't very long-lived. They aren't those big roots you see when you dig into the soil. They're mostly the fine roots. So this rhizosphere is ends up being a leached zone. It's more weathered. It's got lots of carbon material in it. But a lot of that carbon gets recycled by the microbes. But we have hot spots of carbon, roots moving through the soil. And these are scanning electron microscope images. I'm going to show you a couple of these. This is, again, from the Santa Cruz soil. Down here is a root. This is its former zone. It pulled up through here. 500 microns is what this bar is. That's a half a millimeter wide. So 1 inch has 25,000 or so microns. So the electron microscope allows us to get really close images. Here's our rhizosphere. These are, I think, I'm not 100% sure, but I think these are some kind of armored amoeba that lived in the root zone. And then up here is what is a biofilm. This is microbes, and we're going to focus in on that right now. There it is again. Here's the root zone. This is the biofilm. It's hard to kind of tell. You have a little look at it, and we're now down at 50 microns. Oh, let me just say, it's actinomycetes, common soil bacteria. But when I first saw it, I didn't even know that name. I just thought, my God, I have SpaghettiOs in my soil. Here we are at 5 microns and 10 microns. Actinomycetes are apparently very good at decaying cellulose and chitin, which are particularly hard to break down. And I've also learned on the internet that it's what gives the soil that lovely smell when you break something open. You're smelling the actinomycetes. And here they are right inside our rhizosphere. So we have root exudates reacting with the soil and releasing nutrients. We're stimulating symbiotic microbes, and that's causing that, releasing the nutrients, and the plants get the nutrients, the soil nutrients they need. And basically, the plants are digesting the soil. They're digesting the parent material and turning it into soil. And you can kind of see that here. Here's our root. Here's some clay. This leached zone here is has less iron, and the iron is still out here. So this gets me to like, what does a preserved rhizosphere look like? What would happen if I had rhizospheres that formed and didn't get destroyed? Well, we would have root exudates that created mineral weathering or leached zones. We would have roots that create carbon-rich zones. And through water uptake, remember when I showed those arrows going in, creating the leach zone? The roots take up the nutrients they need, and they leave behind what they don't need, aluminum and silica mostly, and aluminum and silica form clay minerals. So what we would have in that zone would be precipitation of clay minerals. Okay, and what we did is we separated out these soils into their different colors, did some chemistry and mineralogy on them to see if we could verify some of these guesses of what a rhizosphere would look like. So here's that data. Weight percent iron across the bottom. So this is the concentration of iron in that element. The orange ones are the orange models. The white ones are these white models, and the gray ones are the gray models. Aluminum is high in the gray. That's because it has more clay. I'm going to show you that later. Iron is higher in the orange. Like we could visually see that. And that gray clay has more carbon. So it's kind of showing us what we thought we might see. Here's another way of looking at this. Is X-ray diffraction mineralogy. So these are our total clay minerals and our total primary minerals. And you see all the orange and the white zones still have a lot of primary minerals. The gray has more carbon. And a different way of plotting that, now it's on soil depth. And so you can see that we sampled at different depths on different terraces. But in each depth, the gray zone is higher in carbon than the associated white and orange zone. And when you do an electron microscope image of those clays, they're really fine-grained. This is now 1 micron. These are smaller than 1 micron. And when you have really fine-grained crystals, it tells you that things precipitated quickly. They didn't have time to make nice big crystals. And so this is a hint that this is precipitating fairly quickly. So we know that the roots follow the fractures in the soil. They go to the path of least resistance. And if you look into the literature, you'll see papers that say, oh, the roots are following, new roots follow old root tracks, and there's nutrients there. Here is the edge of a fracture from Terrace 1, and here's some fractures from Terrace 5. I hope you guys can see. Yeah, it shows up pretty good there. You can see the roots along these fractures. Here you can see there's not much clay, but there's organic material. Here, there's a lot of clay. I'm going to show you another picture of this Terrace 5. When you have a soil pit open for a while, the clays will tend to dry out, and if you've ever seen dry clay, it cracks. And you can pull these we call them PEDs. You can pull these things right off the wall of the pit, and they open up along these fractures, and they reveal the roots following this gray clay. That's really pretty cool. So my model of mottling formation. We have all this organic material, and really most of the roots are up here, and we don't see them. Well, why not? Because we have these things. These are gopher holes. We have gophers, we have worms, we have bugs. We have things that are up here churning this high organic soil over, so the rhizosphere is always broken up. It's not preserved. But once you get below the zone of bioturbation, that's what they call it, really. Once you get below that zone, you might expect to see these rhizospheres preserved because they haven't been mixed back up. And that's what we think is going on. The roots, the deeper roots, the ones that are going down to get water, exude their processes, and after 90,000 years, you start to they reinforce each other, and you build up these clay-rich zones in the deep soil. I've got some take-home messages. So we've been on a trip. along the terraces. We might have been in a different place than you planned to go, but marine terraces, remember we started out, they're the result of sea level change and coastal uplift. They're an archive of soil development over time. They're rich in long-term processes that scientists, ecologists, we can all study. Soil chronal sequences are great landscapes to study these long-term processes. The soil carbon distribution and preservation is highly regulated by soil moisture. We saw it in the Mattole soils. And hopefully we can define how that happens in our future work as we synthesize the data that's coming in. Root and rhizosphere processes are important players in deep soil structure, where it's not being bioturbated. Advances in soil carbon cycling are being made by scientists working across disciplines. We need soil scientists to talk to the plant physiologists, to talk to the microbiologists, and the geologists. And E.O. Wilson, I think, said, we know more about the stars than we do about the land under our feet. We really have a lot more to learn out there. And we're lucky to have these places preserved where we can study them. So I want to thank you. I also want to thank the programs which fund our work in the USGS. I need to thank our landowners, the Wilder Ranch State Park and the state parks in general. We've worked in a couple of other places, and the BLM, the Bureau of Land Management, the Lost Coast, which is where and we actually have some private landowners that allow us access to their land that I really want to thank. And then there's even more colleagues. Digging these soil pits takes a lot of people and a lot of work. And if you're really interested in some of these things that people have discovered over the last 20 years, I've got a couple of good books up here to get you into the soil processes. And with that, I thank you for your attention, and I guess we'll take questions.
Thank you, Jory, for a great talk. So Jory has time to take some questions and is happy to do that. She is flying out tomorrow. But anyway, for those who came in late, please use the microphones at the back of the room. And if somebody can't get up to a microphone, just raise your hand and I'll bring the microphone to you. And also for those who came in late, there are flyers for next month's talk on the 28th of April on the table in the back corner of the room there. So with that, Jory will be happy to take questions.
Hi.
Hi.
Well, I live in Terrace 1 in Santa Cruz.
You are lucky.
I live in Garden. So the question I have is not about the soil. It's about the terrace I'm living on. We read in the newspaper that the ocean seas are rising. At the same time, there's tectonic activity. So is my house on Terrace 1 going to be flooded or uplifted? But
How low? How close are you to the ocean?
5-minute walk.
You're probably good.
No, I mean, but what's the no? But what?
That's the other thing I didn't mention is our earthquake people use the terraces to talk about, to say things about how earthquake reoccurrence time and uplift rates and things like that. And that's not what I know much about. The undercutting of the terraces is as sea level rises, as the world warms, and it's going to, if we don't do something. Is a problem for people who live really close, and it's happening in Pacifica already. You know that they're losing housing on the edge of that sea cliff, and it's just eating away and being undercut. So that's why I asked, how close are you to that sea cliff? But
I didn't really mean my house.
Right.
More the dynamics of uplifting and are there any thoughts about how that?
I think in our lifetime, I mean, what did Loma Prieta uplifted? 6 feet, maybe in the highest places. So it's, it's punctuated uplift. And the sea level, the projected sea level rises maybe if Antarctica goes, oh God, I don't want to pull the wrong number out of my head, but it's something like 2 meters, 6 feet. Is that right?
IPCC says about a meter. Jim Hansen's group just came out with a paper where they say that's probably the low end could be 6 meters by 2100. That's a matter of hot debate.
Right,
but
yeah.
Definitely, the thing that everybody seems to agree on is sea level is rising.
Is rising, yeah. Did I answer that? Yeah, okay.
Hi. I think this is a plant question, but I'll ask it anyway. I was intrigued by the paths the roots take. They would take the new roots would follow the old roots. But if they're seeking minerals, those old paths are in fact depletion zones.
Right.
So
right, I was confused by
why
that
would
too.
they keep going to the same place, and would they ever branch out to some of the orange areas to pick up new stuff?
I think they do branch out a little bit. I also think it involves their symbiotic fungi, which is like a whole nother talk that I don't really know that much about. But most plants are have obligates. It means they need their symbiotic fungi, and the fungi extend the nutrient uptake ability of the plants. And I kind of view that white zone as the zone where the fungi are going out into the soil more. But I have wrestled with that question. Why are they following that path where there's not as many nutrients? And it may be that I can't answer that entirely.
In Northern California, in the northern parts, like up near Fort Bragg and Mendocino, there's a common growth ecosystem called the pygmy forest.
Right, the pygmy forest, yeah.
They generally lie on those terraces, in
Right,
other words, the flat areas. And could you talk a little bit about what you've studied, or if you've studied those areas, and what you've learned from them?
Right, and I meant to get back to this. When I showed you the picture of with the agriculture on that first terrace, and I talked about nutrient depletion, a lot of those older terraces have so few nutrients that the forests, what grows on them are pygmy forests. And up near Fort Bragg, and there's the Mendocino ecological staircase, but there's actually pygmy forests on not the Mendocino, Monterey Peninsula, high up. It's under golf courses and stuff, but there's still remnants of that. So these older terraces have so few nutrients that the ecosystem is changed into a pygmy. Both. She asked if it was because it was leached or old. Yeah.
I got a question. I have this book called Burns Mystery, and has a picture of Bean Hollow's Pebble Beach, 1890, and it has the sea level right here in the photograph. Here's photographs of this year. Same ocean, same beach, sea level's the same. 126 years difference between this picture and these photographs. So how come the sea level hasn't changed in the last 126 years?
Well, that coast has been uplifted by several earthquakes, one of them being Loma Prieta. And the sea level rise is different in different parts of the world. In what we know, I'm getting to the edge of my knowledge. A lot of the sea level rise that we're seeing now isn't necessarily due to the ice retreat, it's due to the heating of the oceans. As the oceans heat, they are expanding. I want to say on the East Coast, they've had like 8 inches of sea level rise, which is why Hurricane Sandy flooded New York. So it's minimal right now, but we're noticing it.
Yeah, but NASA satellites says that global warming stopped in 1997.
Oh, I don't know about that study, and I really don't want to get into that debate because it's not what I know about. But I thank you for your question.
I have a related question. It had to do with sea level change. You had an interesting chart in the beginning of your talk where you showed the changing sea levels for the last 450,000 years. Mm-hmm. That's a long time.
Yeah.
Where does that data come from?
Oh, God. It's data. If you looked on the right side of the chart where I didn't focus on. it's based on oxygen 18 isotopes of a small sea animal called a foraminifera. And the foraminifera create a calcium carbonate shell. It's like a seashell, but they're microscopic. And they take up, the oxygen isotope tells us something about the sea temperature. And that's about as far as I can take you on that one. But we do know about sea level lowering due to glacial cycles. And they also base similar plots like this on the ice cores that they've taken from Greenland and Antarctica, where they measured different types of gases. So there's different types of the sea level curve based on different data, but they're all they agree with each other pretty well.
Hi again. So you had a slide that showed the difference between the Santa Cruz and the Matoli River, I think it was, and how you had a dashed line, a vertical line that was like fungus and plants for the Matoli, and then bacteria for the.
Right, that would have been the carbon isotopes.
Right.
Yeah.
I'm just curious, can you say more about, I mean, that implies like there's not as much bacteria in the Matoli soils,
Right.
or is that a correct assumption? And can you just say more about why
I think what's happening is because remember, we have very little carbon at Santa Cruz, so that these microbes, I'm going to have to see it, I'm going to have to do this. The microbes don't have a lot to work with, and so they reprocess it over and over again. And so here they're obviously working. There's so much carbon there. There's going to be a ton of microbiology there. We do have people measuring the microbiology. Don't have any of that data back yet, but I think that there's just so much carbon here it swamps that signal. Whereas we're sort of carbon starved, and so that forces these. Where were those? Oh, it was the carbon-nitrogen ratio, right? Yeah. So it forces these microbes here to just recycle that organic matter, and it moves that carbon more towards the microbial end of things, the bacterial end of things. Does that make sense?
Yeah, thanks.
Okay, great. Well, thank you. You guys have been one more question?
So how long does it take you to get convinced the BLM or Wildlife Ranch that you're good guys and won't do anything stupid? And what do you do with the hole when it's all done? Do you fill it in, or you?
Oh
save it
yeah,
for
we fill it in. Well,
traps?
actually, we have samplers in them that measure temperature and moisture and things like that, and we go up and then read them off of the data off. BLM has been really good to us. They've allowed us access, and I haven't had any problems.
Just one sort of out-of-the-box question. If money were no object and numbers of assistants to do things was no object, seriously, I'm sure you've thought about this. If you had 2 to 3 years, what would you want to do to answer what question with that kind of those kinds of resources?
Well, we'd like to put in lots of CO2 analyzers to get what the CO2 is in the soil. And we're sort of heading on that, but we do it by our bootstraps. We're temperature probes, moisture probes, water in, water out, soil water samplers, where we can measure the dissolved organic matter, LIDAR, which is a radar of the actual surfaces, plant surveys. Oh, I could go on and on.
I know that some of that would require some equipment and skilled training, but is anything that you just described suitable for the citizen science kinds of projects?
Oh, yeah. Oh. You know, in Matole, and part of the reason I didn't have those nice water plots for Matole is that we just don't get there often enough because it's an 8-hour drive. And so if we got citizens involved up there who could come and read our samplers, once a month or something, that's the kind of stuff that would be really helpful. But that it's a long way from anywhere. But we have thought about citizen science help to some extent, and some of this stuff could be amenable for that. I thank you for the suggestion.
I don't know if it's okay to ask another question, but as the soil is uplifted, have ecologists been able to work out an ecological succession sequence from the coast?
There's been a lot of, especially up at places like Jug Handle,
Right.
where they have the ecological staircase. There has been a lot of work on ecological succession on these chronosequences, and the ecologists actually have their own kind of ecological chronosequence on retreating. Up near Glacier Bay, I know there's been a bunch of work with the retreating glaciers exposing the surface and the plants then, what do you call it, migrating into those new lands. So that, yeah.
Mm-hmm.
So the soil is made by the plants, but then the plants then react to the kind of soil that's there too. So it's
Right.
it's a really interesting dynamic once you start thinking about the feedbacks that have to go on over long periods of time.
Okay, do we know, do we know what plants, well, I guess probably trees and large plants start off in zone 1, and then as we migrate higher up,
Yeah.
do we go to grasses and smaller
When we
plants?
started this work, I thought that these were probably grasslands the whole time because we know that they were coastal prairies when the Europeans arrived. But when you get into some of the firework by, there's a USGS scientist named John Keeley, which I just have recently talked to him, and he said, the prairie grasslands, coastal prairies were maintained by the indigenous people here. And so they've probably only been coastal prairie, God, what did he say? Maybe 12,000 years, 14,000 years. So for not very long. So it's likely that those models I see deep in those old soils were forest or chaparral or something like that. I didn't want to put that in the talk itself because it just complicates things. But I do know that it's not forever been coastal prairie now. Thank you. Well, thanks. You've been a great audience. Appreciate it.